Hello everyone, welcome back to the channel. And today we're going to talk about protein structure. In the previous videos, we did talk about how proteins are built. We know that amino acids are the building blocks of a protein. So the structure of a protein is responsible what that protein will do in the body and what function it will have in the body. So there are four type of structures that we're going to discuss in this lecture. First, we're going to study primary structure. Primary structure is basically amino acid joined by a peptide bond in a linear fashion. So very simple. As you see the structure, these are amino acids right here. So this is a peptide bond in between them. So that's the primary structure basically. And the next, we're going to talk about secondary structure. So secondary structure is it's a three-dimensional structure. There are two type of secondary structures. One is called alpha helix, and second is called beta plated sheets. And alpha helix also called as alpha plated sheets. They have structure like this spiral type of structure. They do have hydrogen bonding between O group and NH group. So that's the characteristics of alpha helix sheets or alpha plated sheets, and it always occurs on the fourth residue. This is a fourth amino acid, so it occurs. So the count starts from this carbon right here. 1, 2, 3, 4. On the fourth residue, it joins with this oxygen right here. So that's called alpha and alpha plated sheets. In beta plated sheets, you have two or more peptide chains. So this one peptide chain running in this direction probably, and there is H bonding between this sheet and this is hydrogen, this one. So that's how these beta plated sheets are. Now there could be a sheet going in this direction too. That could be also part of that structure. So there would be multiple sheets in this fashion. So these are called beta plated sheet. So these are mostly involve small R chain amino acids. If the R chain is big, that would not be a beta plated sheet. That would be a tertiary structure, which we talk about next. So that's the characteristics of a beta plated sheet, that they only involve small R chain amino acids. So now let's take a look of tertiary structure. Now tertiary structure also has a three-dimensional structure, just like secondary structure, but it involves complex R chain amino acids. So that's the difference. So for instance, this whole circle that I just for illustration here, this is a peptide and or protein, Now it has a polar groups outside reacting with the aqueous environment, and sometimes they also make bond with each other, this NH3 group and COO group. So these are polar groups outside the structure of a protein. So this is all protein structure right here, this round shape. And non-polar groups, like we talked about before, they kind of inside right here. These are non-polar groups. They're hydrophobic. They stay away from water. So these are the four characteristics of a tertiary structure, that they are three-dimensional. They involve complex R chain. Polar groups are outside, non-polar groups are inside. And they do also have a disulfide bond between cysteine group. So somewhere in between here, there are many cysteine groups that would have this type of connection between them, and which help maintain the structure of the overall protein. That's disulfide bond is been known for. So remember these four properties of tertiary structure. Now we can talk about quaternary structure. Now this whole 4 units that you see is one protein. This is all one protein. This is all one protein. And these are just the subunit of this protein. So like this definition is, it is formed when two or more folded proteins, see they are already folded, folded peptide chains come together to make one complex structure, which is this protein. So that's a quaternary structure. So in this video, I will just summarize it, what we talked about. We did talk about that protein structure is responsible, that make sure what function that protein is going to do in the body. So there's a primary structure in a linear shape. Secondary structure has two types of structure, alpha helix, alpha plated sheets, and beta plated sheets. Third one is a tertiary structure. It has a polar groups outside, non-polar groups inside, and disulfide bound to stability. And quaternary structure. Folded proteins come together and make a big protein. And that's it for all four structure, guys. If you like the video, go ahead and subscribe the channel. And in the next channel, we'll talk more of these good ideas. Till then, have a good day.